You're live now.
Lovely. So, hello everyone. My name is Aisha. I'm a lecturer at the Division of Pharmacy and Optometry, and I've been part of University of Manchester since 2010. So I'm very passionate about public engagement and for sharing scientific advancements with public, and therefore I've been involved in several public engagement activities and events, including co-organizing Cafe Scientific talks since 2015. And just only a few weeks ago, we're really excited to having rebranded ourselves as Live with Scientists. So as part of this initiative, as part of Live with Scientists, we have been reaching out to bring you blog articles, question and answer pieces, and live talks. And today we will be hosting a scientist from University of Manchester. So firstly, I would like to thank you all for your engagement with our materials so far and for joining us today. And I also would like to thank to our team of volunteers who have been working really hard towards this initiative. So please keep following us through our website, through our mailing list and social media accounts on Facebook, Instagram and Twitter. So for our talk today, it is an absolute pleasure to have and to introduce Lynda Harris, Dr. Lynda Harris to you. Lynda is a senior lecturer at Division of Pharmacy and Optometry at the University of Manchester. She mainly focuses on placental research and uses nanomedicines as a tool in her research approaches. So as our format, Lynda will start by giving a talk for 45 minutes, introducing her research to us. And then we will have a 10-minute break to give you a chance to submit your questions using the YouTube comment box or other channels, through our social accounts, social media accounts and website. After this break, we will resume to answer your questions. So, Lynda, we look forward to hearing about your research, and thanks again for joining us today.
Okay, thank you, Aisha, for that really kind introduction. What I'm going to do now is share my screen, so hopefully you'll be able to see my presentation. Aisha, are you able to confirm that you can see my screen now, or Scott?
I think.
Yeah, that's fine.
Okay, great. So thank you again, Aisha, for a really nice introduction to the presentation. What I'm going to tell you about today is how my lab are using nanomedicines to create safer medicines for use in pregnancy. So before I start, I'm just giving an outline I'm planning to give. Oh, there we go. So firstly, I'm just going to spend a couple of minutes explaining who I am and how I ended up working in this area of research. I'm then going to answer a series of questions. The first one being, why do we actually need new medicines for treating pregnant women? And to answer these questions, we're going to explore the different pregnancy complications that can arise and the role of the placenta in pregnancy complications. I'm then going to discuss why it's so difficult to make new medicines for pregnant women, and how we can address some of the issues around safety of designing new medicines for use in pregnancy. And what I've done to address this is to follow developments in the field of cancer biology and use technology which is known as targeted drug delivery, which will help us to develop new medicines that are much more suitable for use in pregnant women. Then taking this forward, I'm going to talk more about how we can use these nanomedicines specifically to deliver drugs to the placenta. I'm going to talk about some work we've done in the lab of how we developed these new drug delivery systems and how we showed they actually worked, and then tell you what my lab is up to next. So before I start the talk, I just want to flag up a couple of things. Because we're talking about pregnancy complications, I will make a very brief mention of miscarriage and stillbirth. I'm also going to show a few images of human placentas. They are historical images. I don't think there's anything there too graphic, but I just wanted to tell you that they will be included. So firstly, who am I? Well, I was mostly raised in Yorkshire. I was born in Scunthorpe and raised in Sheffield. I was interested in biology and biomedical science for a young age, so I went to the University of Reading to do my degree there. So I got a BSc in pathobiology, which is equivalent to the modern day biomedical science courses. And I got really interested in blood vessels and blood vessel function. So I ended up doing a PhD there, which looked at how blood vessels remodel in the process of heart disease and some of the changes you see in the structure of the blood vessels. Once you've got your PhD, you then go and work as a postdoctoral scientist in the lab of another scientist. So I was looking for projects to go and work on, applying for jobs, and I saw a really interesting job advertised in Manchester, which was looking at how blood vessels remodel in the womb in pregnancy and how the placenta interacts with the blood vessels in the womb. So in 2003, I moved back up north, admittedly to the wrong side of the Pennines this time, but started working as a postdoctoral scientist at the University of Manchester. So you start working in pregnancy and inevitably you get involved in working in placental biology. And this really caught my attention. I didn't feel that there was enough people researching this really important area. So this is where I decided I wanted to focus my academic career. So to move forward in academia, what you start to do after you've trained in someone else's lab is to try and think of ideas and get funding to start your own lab. So it seemed evident to me that we know a lot about pregnancy complications, we know about what goes wrong, but there was this real lack of knowledge of developing drugs and treatments for pregnant women. So I applied for some money and was awarded a 5-year fellowship to start my own lab to develop new medicines for use in pregnancy. I was really lucky because the first year of this fellowship allowed me to go and train in the US. So I went to the University of California, Santa Barbara for a year, and I worked in this beautiful lab here, which had a nice sea view. This was working with a very eminent cancer biologist called Professor Ruslati, who worked on targeted delivery of drugs to tumours. So I learnt a lot of his technology, brought it back to Manchester, and set up my lab group there at the beginning of 2012. At that point, I'd been in the Maternal and Fetal Health Research Centre, but in 2015, I moved to the School of Pharmacy and was appointed as lecturer there, and then was promoted to senior lecturer in 2018. So I'm now split between the two departments, and my lab broadly focuses on developing new treatments for pregnancy complications. So why do we need medicines for use in pregnancy? Well, there are major complications in pregnancy, and I'm sure if you think of sisters, friends, daughters, I'm sure you can all think of somebody who had a baby that was born too small or too early, and potentially people who have a miscarriage as well. So there are quite a lot of different complications in pregnancy that affect a large number of pregnant women. So babies that are born too early are considered to be born preterm, so anything before 37 weeks of pregnancy. Normal term pregnancy is 40 weeks, so this affects about 8% of births. Fetal growth restriction, where the baby is not reaching its full growth potential and it's considered small, this can affect 5 to 10% of pregnancies. Preeclampsia is a condition where the mother's blood pressure gets dangerously high and her kidneys start to fail, and also there's risks for the baby there. So this condition also affects between 5 and 10% of pregnancies. Also, one issue is having babies that grow too big. So the condition macrosomia describes babies that are larger than 4 kilos, and it can occur often in women with diabetes or women who develop diabetes in pregnancy. So there's a high blood sugar level, the baby's getting more nutrients than it would normally receive, so it grows too big. You might not think this is an issue, but actually when it comes to delivering the baby, it can be difficult to have a normal vaginal delivery, and the women may either experience damage or they may need an emergency cesarean section. And also miscarriage and stillbirth, so 1 in 4 women will experience a miscarriage in their lifetime. And even in the UK, 1 in 200 babies are born stillborn. So a miscarriage is considered a pregnancy loss up to 23 weeks of pregnancy, and a stillbirth is a loss of a baby from 24 weeks of pregnancy till term, which is 40 weeks. So I'm sure you can understand there's various short-term consequences of all these conditions. It might be that the baby needs neonatal intensive care, the babies are born too small and too early and are often very sick, and their lungs are not mature enough yet. And this, as well as the physical conditions, can cause a lot of mental distress and anguish for the family. But also, what we know now is if babies are born too small or too early, there's long-term consequences for the health of that child as it goes through its life. So if you are born too small or too early, you are actually at increased risk of developing a number of different conditions, such as heart disease, type 2 diabetes, high blood pressure, or having high cholesterol, and other things like you're at a slightly increased risk of developing schizophrenia or osteoporosis. So actually, if we can improve health in pregnancy and make sure as many babies are born healthy as we can, as well as reducing the short-term consequences, we can ensure more babies have good health and through adulthood. And this is why I'm interested in the placenta. So what is the placenta? The placenta is an organ that develops alongside the baby in the womb. And looking at the image here, you can see that it's intimately associated with the wall of the womb. So it's not just a free-floating organ that's in the womb with the baby. It's really strongly adhered to the wall of the womb, and it's responsible for transporting all the nutrients and oxygen that the developing baby needs. So if we want a healthy baby, we need to have a healthy placenta. A normal placenta weighs about 500 grams. It's about the size of a dinner plate, so about 22 centimetres in diameter. And everything that the placenta does will control the growth of the baby. So this image here I quite like. So you have the placenta here, and looking closely you can see all the different nutrients that are transported by the placenta. So that might be the macronutrients and the micronutrients to make sure the baby has everything it needs to grow and survive. We also know that baby needs oxygen. So the placenta, as well as acting as an intestine, I guess, to transfer nutrients to the baby, it can also transfer oxygen, so it's behaving like a lung. It can also remove waste as well from the baby's circulation. So they've got a role for the placenta in nutrient and oxygen transport. It also produces hormones as well, so it can produce things like progesterone and estrogen to maintain the pregnancy, and also HCG, so that's human chorionic gonadotrophin, so that is the hormone that is tested by the pregnancy test. I mentioned it has a role in waste removal, but it also acts as a barrier both to infectious agents, so it prevents many bacterial and viral infections crossing from the mother's circulation to the babies, but it will also prevent the transfer of toxic substances. So these might be drugs or any other agents that might be harmful to the baby. So the placenta has very many roles, all of which that supporting the growth of the baby. So here we've got the basic structure of the placenta. On the left-hand side here, we've got a placenta from 9 weeks of pregnancy. And on the right, you've got a placenta from 40 weeks, which is term, end of pregnancy. So what you can first notice about this is that during the first stages of pregnancy is the placenta grows much, much faster than that of the baby. This is because, again, it's the placenta that supports the growth of the baby. So the placenta needs to be large and functional so it can transport enough nutrients to support and maintain the growth of the baby. By 40 weeks, obviously, the baby has outgrown the size of the placenta. But considering the structure, the placenta, if you look at it under a microscope, looks a lot like seaweed. So it has lots of finger-like projections, which helps it to have a large surface area for transporting nutrients. And what happens if we look at it under the placenta? This image here is a section through the placenta. So if you imagine this is a section through one of those finger-like projections, the placenta is made up of what we call trophoblast cells. So placental cells, the scientific name for those is the trophoblast. So they're shown in this image here in pink. They actually form a double layer or a bilayer to protect the underlying cell types. So this trophoblast layer is the specialist layer in the placenta. This is the one that's responsible for transporting the nutrients, for producing the hormones, for excreting the waste. And this is the one that's important for healthy pregnancy. What I've also got here is a picture of a mouse placenta. So I'm going to be showing you some pictures of mice placentas a bit further on in the talk, so I've included this here just to explain the structure. On the right hand side, this is a mouse fetus just prior to delivery. Mice have fetuses in the uterus for between about 18 and 19 days. And you can see here each mouse fetus has its own placenta, which is this button-like structure just on the top of the fetus, which is about 1 centimetre across. So if you were to cut through the mouse placenta, the top half of this middle image is the maternal facing, or the part of the placenta that attaches to the maternal wall of the uterus. And within it, you can see the spiral arteries. So these are the blood vessels that supply the placenta with the mother's blood. Then you have the labyrinth layer, which is a meshwork of both the maternal and the fetal blood vessels. So this is the area in the mouse placenta where nutrient exchange takes place. So if we were trying to deliver nutrients from the mother's blood to the mouse fetus, the blood would come through the spiral arteries and then feed into this labyrinth, and the nutrients would be exchanged from the maternal vascular, or the maternal blood vessels, to the fetal blood vessels. Similarly, if you wanted to deliver drugs this way, they would come in down the spiral arteries and cross the labyrinth. So what can go wrong? Well, what we have learnt over many years of research is that many of the pregnancy complications that we see in the clinic are linked to poor placental growth or poor placental function. So if the placenta is working well, then this is great. This means you've got a healthy baby. But as soon as the placenta doesn't grow properly or doesn't work properly, this means we've got a smaller baby that's not going to do so well and is at risk of complications. So there's various things that can be wrong with a placenta. Simply, a placenta can be too small. So if it's too small, that means it's not very efficient at transporting nutrients to the baby. So it might be that the placenta is working well, there might be a high concentration of nutrients in the mother's blood. But if you think of it like a sponge, if you've got a small sponge and you're trying to mop up a spill on the worktop, there's only so much liquid you can mop up. And it's with a small placenta, there's only so much of those nutrients you can transfer to the baby. But if you've got a big sponge or a big placenta, that can really efficiently mop up nutrients from the mother's blood and transfer them to the baby. It might also be that the placenta itself is bad at transporting certain nutrients, so that the placenta has a series of different nutrient transporters on the surface that allow it to grab those nutrients from the mother's blood, transport them across the placenta to the baby. But if you haven't got enough of those specific nutrient transporters, even if you've got a big placenta, a big surface area, and even if you've got a lot of nutrients within the mother's blood, there's just not enough of these transporters to be able to supply the nutrients to the baby at a rate that will maintain its growth. So in this case, again, the baby's growth will slow down. We also see pregnancies where the blood flow through the womb is not efficient. So in normal pregnancy, the blood vessels of the womb remodel, so they become wider, and this allows a larger volume of blood to be delivered to the baby. However, if this process doesn't occur properly, it means, again, you can't get enough blood to support that developing baby. So again, that means that can restrict the growth of the placenta, it can restrict the growth of the baby, and you can get low oxygen levels, which again can lead to damage to the placenta. And finally, you might get a placenta that's aging prematurely. So what we mean by this is the placenta is pretty much designed to last for 9 months, and then by the time those 9 months are up, it's not working so efficiently anymore. But sometimes placentas can age prematurely, so when you look at them under the microscope, some of the cells are starting to die, there's evidence of damage. So again, perhaps we might need to give treatments to prevent against this premature aging. So what can we do to address one or more of these issues? So if we were a doctor, you might think, well, if a placenta is too small, perhaps we can give it some drugs that might promote placental growth so it becomes a normal size. If it was bad at transporting nutrients, well, we just give some drugs that we know could promote nutrient exchange. If it's not receiving enough blood, we could give drugs to increase the blood flow, because there's various drugs available for other conditions that we know can relax blood vessels and increase blood flow. Or if the placenta is aging prematurely, well, maybe we just give drugs to mend the damage. We can have face creams that prevent our face from aging prematurely. So why can't we have drugs to prevent our placenta from aging prematurely? So this is all well and good, and actually in the lab, this is what we can do. The issue comes when we're actually thinking about developing and testing medicines for pregnant women, which is what I'm going to talk about next. So why is it so difficult to make new medicines for pregnant women? Well, one of the major issues that's had long-standing effects on the pharmaceutical companies' interest in this area was the thalidomide scandal of the late 1950s and early 1960s. So thalidomide was a drug that was developed in that time, and it was found to be really effective at preventing morning sickness. So it was prescribed to very many pregnant women, and unfortunately, when the women had taken that drug, many of their babies were born with quite severe deformities of the limbs. So there were over 10,000 thalidomide babies born. So this was a big shock to the pharmaceutical industry. They'd created a drug that had actually harmed someone. And as well as that, it cost them a lot of money in compensation and lawsuits. So this was really damaging to them. And their response was, well, pregnant women are a high-risk group. We don't want to make any more medicines for them because it's too risky for us, and we don't want to have a similar scandal. So actually, there are very, very few medicines available for use in pregnancy, and certainly there are no medicines available for treating conditions such as preeclampsia or fetal growth restriction, which were the conditions that I mentioned earlier. Another issue is that women are almost never included in clinical trials. So the reason for that is highlighted really nicely in this cartoon. So the doctor's saying, if we don't know which drugs are safest and effective for pregnant women, the woman is saying, if we don't know which drugs are safe and effective, why don't they just let more of us into clinical trials? And the doctor's saying, well, we need to protect you from all those untested drugs. So if we protect the women by not including them in the trials, we never get that really important information that tells us which drugs are safe and which drugs are not. As well as this, in pregnancy, a woman's body changes quite dramatically. So her heart rate increases, her blood pressure is reduced in the first half of pregnancy, and then is elevated back up in the second half. Her heart works harder. her liver and hormones, or liver function changes, kidney function changes, levels of hormones change. So actually, if you give a drug to a pregnant woman, that drug might behave differently to a non-pregnant woman. So again, if you include a few pregnant women in your trial, you might get skewed results. So we know that some drugs are metabolized more quickly in pregnant women than non-pregnant women. We know some are metabolized less quickly. And as in pregnancy you lay down a lot of fat, often you can get drugs that will accumulate in the fat within your body. So this is another reason, because pregnant women behave very differently, they don't include them in clinical trials. The outcome is, for all but the most basic of medicines, women are generally encouraged not to take anything at all. There are no treatments for pregnancy complications, so all you can do is monitor the growth of the baby, and if the baby appears not to be growing or is in distress, then you just deliver the baby. But how can we address this problem? What can we do to try and reduce some of the risks associated with making drugs for use in pregnancy? How can we prevent drugs crossing to the fetus and causing abnormalities? Is there something we can do to address this? Well, yes, it turns out there is. So maybe 20 years ago, similar questions were being asked with regard to tumour drug delivery. So if you have a tumour, you're often given chemotherapy, and the way chemotherapy works is that it's toxic and it will kill the tumour cells. But the problem is, if you administer this to the body, you can get very serious side effects. So as well as killing the tumour, it will also be damaging all different types of tissues in your body. So the cancer biologists were thinking, well, is there a way that we can reduce these side effects for patients and see if we can get more of these cytotoxic tumour drugs delivered to the tumour and getting less side effects in the body? So they developed ways of being able to make their drugs so that they only targeted the tumour. So they can design the packaging for the drug that's when it's ingested, it accumulates in the tumour and avoids all the other areas within the body. So that seemed like a really good way to minimise side effects and minimise risk if you know exactly where that drug's going to go. So when I was thinking about ideas for my fellowship funding and what kind of projects should I be working on to advance the field, I wondered whether we could take some of the lessons learned for delivering drugs directly to tumours and deliver them directly to the placenta. And indeed, when I went and spent my year in California working in the big drug delivery lab there, that is what I learned how to do. So how do we do it? Well, I've got an analogy here. So if I wanted to send a Valentine to my sweetheart, but I didn't know where they lived, one thing I could do is send maybe 500,000 Valentine's cards, one address to each house in Greater Manchester, and just hope that one turned up at the right address. The other thing is, if I know the address of my sweetheart, all I need to do is send one Valentine with that address and that postcode on there, and I know that the card will get to the right place. This is kind of what we're doing with drug delivery. So if you imagine, if you've got a headache or a sore back and you take a painkiller, what you're doing is you're taking a tablet, and those drug particles, when they get into your stomach and then into your bloodstream, in the blood, they go to every single area of your body, and you're just hoping that they'll get to your head and they'll treat your headache, or they'll get to your back and treat your backache. But it's not very efficient, because only a small proportion of the drug is actually treating your headache, because some of it is in your knees, some of it's in your liver, some of it's in your kidneys, all circulating in the blood. So to achieve targeted drug delivery and send a drug just to your head or just to your back or just to your placenta, what we need to do is we need to know the address of the destination. So targeted drug delivery is about selectively delivering a drug only to its site of action, not to any other cells or tissues. And to do this, we need the address of the organ or tissue of interest. So how does that work? Well, actually, the more we know about the body, we know that different cell types and different organs and different blood vessels are all unique. On the surface of each cell and each tissue, you get lots of different molecules displayed. And we know that some of those molecules are expressed, the same ones are expressed on different organs and different tissues. But what we've come to learn in recent years is actually if you pick out a specific organ or a specific cell type, there's usually always one or two molecules that are expressed on that cell or that blood vessel or that organ that are unique to that site and are not expressed anywhere else. So we can refer to this as the address or the postcode. So if we can find out what those molecules are, then that will allow us to address our drugs to that location so we can get targeted drug delivery. What we actually do is we do literally hide a drug in some kind of envelope. So if the drug is free, it can interact with any tissue, any cell or any organ it comes into contact with. But if we can hide it in some kind of envelope or package, it can't interact with a certain cell type. It can't be taken up by cells. So it will only act at its final destination where its surface address or postcode is telling it where to go. So this has a benefit of having fewer side effects and being able to control exactly where that drug goes. Even better, less drug is needed overall because the treatment is going exactly where you want it to go, straight to its site of action. You're not losing some of it in the other organs. So you can administer less drug into the bloodstream, but achieve a higher local concentration at your site of interest. So then the next question is, what's the postcode of the placenta? If we want to send things to the placenta, how do we know where to send them? So this was the work that I did in the lab in the US. And I'll try to explain how I did that. So if you want to discover a particular postcode, what you have to do is screen all the postcodes and see which ones end up in the right place. The way we do this scientifically is using a viral library. So viruses are very small particles. So if you have a drop of fluid that's got 10 or 100 billion viruses in it, that's actually a very small volume of liquid. So I was able to use a virus that was engineered for screening of short peptides or short molecules. So these viruses, they didn't cause infection. All they could do is replicate. And they were engineered so they randomly expressed a single peptide sequence on their surface. So a peptide is like a small piece of a protein or a chain of amino acids, which are the building blocks of proteins. So it was basically a string of 7 to 10 amino acids displayed on the surface of the virus. So every time the virus replicated, you would get different random sequences expressed. So one virus would only express one peptide sequence, but if you've got 10 billion viruses, you've got 10 billion random sequences within that viral library or that viral pool. So I was able to take some of this library, take a very small amount, 100 microliters, so that's 1/10 of a milliliter, a tiny amount, and within that you've got 10 billion of these viruses. You could take a pregnant mouse and you could inject this viral library into the pregnant mouse and the viruses would circulate around. So remember, those viruses have got those different random postcodes on their surface. So some of those postcodes would bind to the liver, some would bind to the lung, some would bind to the placenta, some would bind to the kidney. But then what you would do is then after you've let the viral library circulate, you then euthanize the mouse, and then you would take out the liver, take out the kidney, or in my case, you would take out the placentas, and then you can grow up the viruses that have bound to the placenta, and then you can sequence them, and that will tell you the postcode or the sequence of the small peptide that was displayed on its surface. So this method allows you to screen billions and billions of random sequences to find the ones that just bind to the placenta. You don't actually do this once, you do this multiple times, because it might be you get a really good sequence that binds to the placenta, but you actually find it binds to the liver as well. So you don't want 50% of your drug going to the liver. So if you repeat this for a few times over, what you find is you get enrichment of those random peptide sequences that bind to the placenta. So that is effectively your placental postcode. So once you know that, you can make your envelope with your placental postcode on the outside and your drug packaged in the middle. So I'm going to show you how we tested how how we determined whether we'd got the right postcode for the mouse placenta. We isolated a variety of these short peptides. They were random sequences of amino acids. They have different names. Usually we named them related to some of the amino acids within their sequence. The one I've worked with quite a lot, we call it, the sequence itself is we call it IRGD, but it's actually a 9 amino acid long sequence. The FAM part here is a fluorescent molecule. So to test whether these peptides bind to the placenta, we synthesize a single peptide and we put a green FAM fluorescent marker on it. You can inject those into the bloodstream of a pregnant mouse and then collect the tissue and see where the fluorescent signal is. So if you've got fluorescent signal in your tissue, that means you've got binding of your peptide postcode. So I've put this picture up here to remind you of the structure of the mouse placenta. So the labyrinth is the area of nutrient exchange, and actually looking on the left-hand side, these are pictures of mouse placenta. The blue staining is just the nuclei within the tissue, and the green staining is that fluorescence coming from the placental peptide. So you can see we got lovely accumulation within the labyrinth of the placentas, and we got lovely accumulation in the spiral arteries that supply blood into the placenta. So we were really happy. This was one of the peptides that worked best. And then, as well as checking the placenta, it was important to check the maternal organs to make sure we were just getting fluorescence and delivery in the placenta and nowhere else. And looking here, we've got the brain, the heart, the kidney, the liver and the spleen, and you can see that there's almost no fluorescence there. So we were confident that this postcode preferentially accumulated in the placenta. We knew it bound to mouse tissue, but what's more important is, does it bind to human placental tissue? So a really basic assay, we took some small pieces of human placenta shown here. So we had some early placental tissue from the first trimester of pregnancy and some term placenta straight after delivery. We cut it up into tiny pieces, dunked it in a solution of our FAM-IRGD, so our postcode molecule with our fluorescent dye attached. And over time, so from 0 hours, 30 minutes, 1 hour, 3 hours, we saw a beautiful accumulation of our placental homing peptide within this outer trophoblast layer. So it worked in first trimester placenta and it worked in term placenta as well. And there's just this image to remind you what the human placenta looks like. And these are the trophoblast cells that we're trying to target. So this is where we would want to deliver drugs to fix the problems. And this is exactly where our postcode molecule was binding. So that was fantastic. So we've got the postcode. Now, how do we make the envelope? What are we going to package our drugs inside? So this is where the nanomedicine aspect comes in. So nanoparticle is any particle that is considered to be between 1 and 100 nanometers in size. Sometimes that range can be included up to 500 nanometers, but broadly between 1 and 100 nanometers in size is considered a nanoparticle. So anything that small, you can call it a nanoparticle. Doesn't matter what it's made of. So if they're treating tumours, often people would use metallic nanoparticles, so iron oxide, gold, or things like silica or carbon. The reason these are used for treating tumours is because they're toxic. They build up in cells and they kill cells. If we're treating a placenta, we don't want to kill it, we want to make it work better. So it's important for us to choose a nanoparticle that's biocompatible, non-toxic, biodegradable. So what kind of nanoparticles could we use? Often you can make them out of polymers, naturally occurring polymers that are non-toxic. You can make them out of fats or lipids. So liposomes are organic nanoparticles that are made of fats. Or cubosomes, these are a liquid crystal, so a similar structure to you'd find in your television screen. So rather than having a single hollow component within inside, it's got multiple compartments within it. And dendrimers are again made from polymers, so they've got long tails which you can package drugs within these complex tangled molecules. So once we've identified the postcode and picked what envelope we're going to use, what we want to know is if we put the postcode on the envelope, does it still go to the placenta? Is it still delivered to the right place? In the lab in the US, they were mainly working on cancer. They had these iron oxide nanoworms. So these are strings of iron oxide nanoparticles that were about 30 nanometers in diameter, and they were decorated again with a fluorescent molecule so you could see them. So what I did, although this wouldn't necessarily be used clinically, just to confirm that if you put my postcode molecule on some kind of nanoparticle envelope, would it end up going to the right place? So again, we've got some images of mouse placenta here that have been injected with these iron oxide nanoworms decorated with the fluorescent postcode peptide. And you see exactly the same localization, beautiful accumulation in the labyrinth area, and also in these maternal spiral arteries that supply blood to the placenta. This end image here is a placenta or a mouse or placenta from a mouse that was injected with iron oxide nanoworms. They had the fluorescent marker, but they lacked the postcode. And you can see really nicely there were no nanoworms accumulating in this placenta. So we know that it's the postcode is ensuring we've got specific targeted delivery to the placenta, but you're not just getting passive accumulation without that postcode there. We also tried some fat or lipid-based nanoparticles. These were very small, again, attached the placental postcode to those, and saw again similar localization to the spiral arteries. So using two different types of envelope and the same postcode, we got placental localization, which was great. So the final design for the nanomedicine, we decided to use liposomes. If we want to get this drug into the clinic as soon as possible, we wanted to use a type of nanoparticle that's already clinically licensed for other conditions. So liposomes, which are just small lipid bubbles made of fat, are used for various medicines already. So we used the formulation, the same composition of fats to make these hollow lipid bubbles. We incorporated a compound called PEG, or polyethylene glycol, which has the ability to protect these nanoparticles from the immune system. So it's regarded as a stealth layer because it allows these nanoparticles to circulate longer in the body, not be detected by the immune cells, and not be gobbled up or excreted. We then put a maleimide group on here, which allowed us to easily couple our postcode molecule to the surface. You have sometimes put a second fluorescent marker inside, but what we're really interested in is this is where the drug would go. So the drug's hidden away so it can be targeted to its site of action. And broadly, these end nanoparticles were between 150 and 200 nanometers in size. Size is often charge dependent, so depending on what drug was packaged inside, the size changed slightly. So just to finish with telling you about one study about how we used these nanoparticles to improve placental blood supply. So what we have here is a molecule called Se175, and we knew that this had previously been used in a few different studies to relax blood vessels. So I mentioned at the start of the talk that we can have issues in pregnancy if the blood supply to the womb is not efficient. So if we were to deliver a drug that relaxed the blood vessels in the womb, potentially we could get improved blood flow, enhanced delivery of oxygen and nutrients to the placenta. This drug had never been tested on uterine or blood vessels before, so before we wanted to encapsulate it into our nanoparticles, we wanted to make sure it worked. The graph you see below shows a measure of blood vessels and their relaxation. So here, this is some uterine or womb arteries taken from a pregnant mouse, and they are constricted, so they're held under tension. So you've got the control blood vessel here holds its tension, and then the axis shows it's measuring constriction and relaxation. So you can see once we applied our Se175 drug just on its own, not in nanoparticles, we saw a really nice decrease in constriction, so indicative of that blood vessel relaxing as you increase the concentration of this drug. So the drug works on mouse uterine arteries in the lab. That's great. What happens if we target this drug to the uterus in a pregnant mouse? So we packaged our Se175 drug into our nanoparticle and put one of our peptides on the surface. So this was a different sequence this time. This is a peptide we call NKG, but this was one that binds very well to uterine blood vessels. Then we used a mouse model that has a phenotype or a disease-like state that's similar to preeclampsia. So it has high blood pressure before pregnancy, but when the mouse gets pregnant, its blood pressure goes up. The babies are born much smaller, and also there's some evidence that the kidneys are not working properly. What we do know is that these mice don't have very good blood flow in the uterus. So what we tried to do is we had different groups of mice that we administered our treatment to. So these are specially bred. They're called eNOS knockout. They're missing the endothelial nitric oxide synthase gene, which allows them to have the phenotype they have. So for some of these mice, we just injected them with saline to control for the injection that we were giving the other groups of mice. Some of the mice we injected with our liposomes, but these liposomes were empty. They only contained saline. There was no drug. Some of the mice we injected with liposomes that contained our Se175, and some of them we gave the free drug, so not encapsulated. And this was compared to normal wild-type mice, so that didn't have the gene knockout. What you're seeing here is the fetal weights, or the weights of the mouse pups at the end of pregnancy. So this is for a normal mouse, the weight should be about 1.1 to 1.2 grams. In your knockout mouse model, this is greatly reduced. So we saw that injection with saline or with liposomes containing saline, it didn't really have any effect on the average weight of those pups. However, what we did see is when we administered the targeted Se175 drug, we did see a significant increase in the mean weight of the mouse fetuses, or the mouse pups, compared to the untreated mice. And actually what we're really interested in on this graph is these pups down here. So in most of these groups, there was a range from big pups, medium-sized pups and small pups, but actually in the group treated with the targeted liposomes, there were no small pups. So we seem to be rescuing those, making them grow bigger, but there were no oversized pups. So we were making the normal ones grow too big. So this is one of the examples where we've used our targeted liposomes to selectively deliver a drug to the uterus of pregnant mice, where we've seen a significant increase in the weight of those mouse pups. So what am I doing now? What's my lab doing now? Well, now we've got this really interesting technology. We're trying different types of drugs to correct some of the different things that might go wrong with placentas. So we might be trying to increase placental growth, protect against aging, again, as I mentioned, improve blood flow to the womb, protect against some of the cell death we see. But also we're exploring different types of nanoparticles, so different types of envelopes, as well as using liposomes. We're looking at using cubosomes. And we're also thinking about might we want to restrict some drugs to the maternal circulation. So what I've told you about today is delivering drugs to the placenta, but there might be some cases where we want to not deliver them to the placenta or the baby, so to keep those drugs within the mother's. So we've been working with some collaborators to develop a certain type of nanoparticle that doesn't bind to the placenta. And overall, the idea is to treat the different types of placental disease we see, and we're learning more and more that actually placental disease isn't one single thing. You might have a small baby, but there might be multiple causes, such as the placenta might be small, or it might lack those nutrient transporters, or it might be suffering from oxidative stress or damage or aging. But the aim is to try and fit the best medicine to the type of placental disease we're seeing. So none of this work could have been done without all my wonderful students, collaborators, and funding from a variety of sources. So my fellowship was from the BBSRC. I'm currently got funding from the MRC and from Tommy's. I've had a number of great students and important mentors through the years, including Professor John Applin in Manchester and Professor Erkki Ruoslahti at the Sanford Burnham Medical Research Institute in Santa Barbara. And I'm currently working with other scientists across the globe to try and maximise this technology.
Wow. So we have got some questions. So I'm going to, first of all, thanks for everyone who has been submitting questions to us during the break. So I'm going to start with one of the first ones that I have received. So are there any genetic conditions determining how much placenta can provide during pregnancy? You know, you mentioned about those 4 categories of problems you can have with the placenta, which might affect the growth of the baby. So could you know, are there any genetic conditions, or is this all environmental?
Good question. So obviously the answer isn't straightforward. So a lot of genetic conditions that lead to pregnancy losses are often associated with abnormalities in the placenta. So yes, that is definitely the case. In cases of Down syndrome, there are abnormalities to the Down syndrome placenta. I'm trying to think what those are at the moment, but I can't bring the details to mind. But yes, that's definitely true. But when we're talking about implantation of that fertilized embryo into the uterus, it's not just the placenta that controls placental growth, it's also what happens in the uterus. So you need a really receptive tissue, you need the immune cells within the uterus to behave in a certain way to allow the placenta to implant and grow properly. So at least some of the issues are caused by how the placenta interacts with the womb. So also the genetic profile of the mother versus the father will determine some of those interactions. So if you remember, the embryo is made up from half the genes of the mother and half from the father. So each embryo will be different, but also the embryo is distinct from the mum, and there's certain particularly related to, immune genes or immune cell genes, the combination of what the mother's genotype and the father's genotype can determine whether the placenta grows really well, or whether the mother mounts a certain response to prevent the placenta from invading and growing too well. So that all ties into it. But yes, it's a complicated area.
Okay. Great, thank you very much. So one of the questions that we get from our viewers today is they're wondering whether the liposomes are leaky at all and how the release of the drugs is triggered.
Yes. Okay. So we can engineer the liposomes depending on the different types of lipids that we use to make them. We can make them very leaky, a little bit leaky, or not leaky at all. So we've used a combination of lipids that we're confident that the drugs are not released. So we can store them in the fridge for 4 to 6 weeks and we get no release of drug. Obviously that's at 4 degrees C. Within our culture systems, we've tested them at 37 degrees C, which is body temperature, and they are also stable. So we're confident that those nanoparticles are not leaky until they're taken up into cells. The placenta is quite a unique organ, so it's quite hard to study exactly how that process takes place. So we don't have the exact details of how those nanoparticles are taken up. But we certainly know that they adhere to the surface of the placenta. There may be a small proportion, if they stay adhered to the surface, that release their drugs on the surface of the placenta. But we've done work where instead of putting a drug in the nanoparticle, you have another fluorescent molecule, so you can track where fluorescence is released. So we are confident that a large proportion of that fluorescence is released inside the cell, so that we're not getting drug leakage.
Okay. So another question from our audience today. So as nanoparticles have larger surface area, and obviously this makes them highly reactive. So how do you prevent the nanoparticles reacting with other substances and causing, preventing them causing further side effects?
So when we've chosen the lipids that we make the liposomes from, they are chosen to be very inert, so that they don't react with other things. Generally, proteins or foreign proteins, foreign peptides can sometimes mount or cause an immune response. So there was a chance that the peptide postcode we were putting on the surface might cause some issues, but in all the safety tests we've done, that doesn't seem to be the case. So the idea is we're making our nanoparticle as safe and as inert as possible, so the body doesn't really see it and doesn't react against it. So we're fairly confident that we're doing that already. Has that answered the question?
I'm going to ask our audience to submit another question if they think this is not answering their question, or if they've got any follow-up questions to follow this up. So they are also asking, are there any side effects of nanoparticles such as liposomes being inserted into the body?
Again, no. So again, it depends on what you make them from. But the choice of lipids or fats are similar to those that are naturally occurring in the body. So no, not with this formulation or composition that we're using. Obviously, if you made them with different things, and if you're using some of the other different types of nanoparticles I mentioned earlier in the talk, yes, definitely. But the idea was to pick something that wouldn't mount a response or cause any damage.
Okay, so from what you're saying, that is it possible to conclude that based on the nanoparticle type, you might get side effects, but for the ones that you're using for pregnancy, this is is unlikely, or
Yes.
very little possibility of happening. But you mentioned earlier that there were other types of nanoparticles, like the ones that are being used in cancer. So if the point of using these particles is, and I know you have got some interest of applying this to cancer studies as well. So if the point of using these nanoparticles is avoiding the side effects, do you think we are actually undoing what we are trying to do if these nanoparticles are going to cause some side effects as well?
No, so I mean, with the idea with cancer is the ultimate aim is to kill the tumour. And if you've got a targeted nanoparticle, you've got a double hit approach. So the chemotherapeutic drug inside the nanoparticle would help kill the tumour, and the design or the composition of the nanoparticle would help kill the tumour. So if you've got it, made it into a targeted nanoparticle which only accumulates in the tumour, then you're fine. It's if you had a nanoparticle that wasn't targeted, where again it might accumulate in different organs in the body, then you would have a double hit of side effects because both the drug and the nanoparticle would be toxic.
Okay. All right, thank you. Well, if you aren't too tired, I've got 4 or 5 more questions that I would like to continue.
Okay.
Yeah, so actually there is this very interesting question. So one of our audience is asking, so how efficient are these medicines for, let's say, or nanomedicines, how useful this nanomedicine approach is if you have got twins and presumably you have got 2 placenta there. Are there any studies showing the efficacy or the penetration of these nanomedicines in each of these placentas?
Okay, that's a good point. So we've not, at the moment, the only human experiments we have done is in placental tissue in the lab, so we've not injected these into humans. However, our mouse studies, mouse pregnancies typically have between 6 and 10 pups per litre. So all our mouse studies have worked really effectively, and we haven't got twins there, we've got between 6 and 10 pups. So as long as you modify the dose, you can imagine if you've got twins, you might need to give double the amount if there's 2 placentas in there. That should work. So yeah, I think that's something that we can easily get around. That shouldn't be an issue for us.
Okay. And so you also mentioned about several physiological changes that the mother has to go through during the pregnancy. Yeah. So again, our audience is wondering, so does the sort of negative changes or underdevelopment of the placenta does actually cause any stress or physiologically any stress or mentally any stress to the patient or the mother is not aware of what's going on until they actually have a scan?
Yes, that person is correct. So in the complication preeclampsia, where the mother's blood pressure gets dangerously high and her kidneys start to fail, we've not, or scientists and medics have not quite completely defined how that occurs, but the main or the current belief is that there are some issues with the placenta not working properly, and you get signals released from the placenta, molecules, because that placenta is either lacking oxygen or has had some kind of damage, and those signals are released into the blood, and they're having effects on the mother, and they're causing her blood pressure to increase and causing her kidneys to fail. So yes, absolutely, that can happen. It doesn't happen in all cases, but yes, definitely.
Okay, great, thank you. So is there any point that you couldn't give the medicine to the patient at all, any time frame, even though you're using a safer version of medicines because you're introducing them as packaged as nanomedicines? Is there any point where you couldn't give this nanomedicine to the patient?
So I think it depends what you're trying to achieve. So at the moment, our aim is to treat the pregnancy complications, and often they will occur in the second half of pregnancy. So end of the second trimester and beginning of the third trimester. So that's when we will be thinking of treating women. If this approach seems to work well in women, we might be able to think about giving preventative treatments, which we might give earlier in pregnancy. So women who have previously had complications or might be considered at high risk of developing them, we could potentially give these medicines to. And as long as we've got all the safety data in pregnancy, that should be fine. One issue, what we might find is that sorry, I'm hearing a bit of an echo. I think, oh, it's gone now. Okay. Some of the postcodes we use also bind to tumours. So when I was in the lab in the US, I screened some of their tumour homing postcodes and found they actually also worked really well on the placenta. So if we used one of these postcodes, we would have to be careful that the woman wasn't diagnosed with a tumour, because what we might find is that some of those nanoparticles went to her previously undiagnosed tumour and some went to the placenta. But we probably wouldn't necessarily use one of those postcodes for our drug if we were going to use it in pregnancy to keep things safe, or we could scan women and just check they hadn't got any undiagnosed tumours prior to giving her that treatment. But that's the main issue we might find.
Great, thank you very much. So one last question though. You mentioned a little bit about this as well, but can you use this technology to avoid conditions related to pregnancy? And can you think of, other than, let's say, preeclampsia, can you think of, or can you give us examples of conditions that nanomedicine treatment can be used for?
So do you mean outside of pregnancy or as preventative?
As a preventative for, let's say, for pregnancy, if you know let's say within 4 weeks of gestation or 6 weeks of gestation, can you screen your patients and determine the ones who might more likely to get, let's say, preeclampsia? Yes. And can you use it for this purpose? And also, what other conditions other than preeclampsia can you actually use nanomedicines for?
Yeah, so definitely. I think just for getting these medicines into the clinic, the fastest way to do that would be to try and treat existing conditions. But yeah, there's no reason why we couldn't package other drugs in the nanoparticles and give them to high-risk women. So you might want to give drugs that improve blood supply to the womb, or give something like antioxidants to protect against oxidative damage, so prevent the placenta from suffering low oxygen. So yes, absolutely, we could do that. So things like fetal growth restriction or preeclampsia or stillbirth as well. We're starting to understand more about what goes wrong with the placenta in stillbirth, and obviously and that could be the result of severe fetal growth restriction. So again, you could give maybe a combination of different drugs this way to try and potentially correct all the things that could go wrong with the placenta, to help someone have a successful pregnancy. Okay,
great. So while you were answering that question, another question has come through, and this is, I think, the final question that I would like to ask without tiring you too much this evening. So our audience is asking, could the postcode have mutations or a chemical change that might affect the health of the mother after being inserted?
Okay, so the idea with the postcode is that it's a very stable structure. We synthesize it in the lab. It's made of a chain of amino acids, so they can't mutate. So that will always stay the same. It can't change. Also, because we are assuming, or the idea is that postcode will take the nanoparticle to the placenta, we're assuming that that's where it will all end up. It won't end up anywhere in any of the other tissues. So as soon as the woman has the baby, she'll then deliver the placenta. The placenta can go for disposal. So there shouldn't be any remaining nanoparticles within her body to cause any kind of toxic effects. So that's a really good point to raise. But also, yeah, the nature of that postcode is that something that is not able to mutate.
Lovely, great. Well, thank you very much for your time, Lynda. It's much appreciated. I really enjoyed it, and I hope our audience did as well.